Hi, my name is Anneka Johnson, and I'm training to become a scientist at Dartmouth College, and I'm so excited to work with you this year. But before we get to do that, I thought I would introduce myself. I'm from a small town in northwest Wisconsin. We only had 1,300 people in our town, and it was so small that I only had 62 people in my graduating class. Growing up in a small town was excellent. We had a local dairy that we could always stop at for ice cream and cheese, and I was able to show our cows, horses, and sheep in the local fair. And I had some of the best teachers. When I was in 7th grade, just like you, I took a class called Crime Scene Investigation with Miss Myers, and we learned how to take fingerprints and use the physics of blood spatter to figure out how far away a drop of blood it fell from. In high school, I had a biology teacher named Mr. Berg, and he taught me about the importance of keeping our rivers and lakes clean. And I even got to help him study freshwater mussels, which are a lot like oysters in the ocean, but they belong in rivers, and they help to filter out the toxins in waters and keep it clean. It was through these experiences that I learned that science could be used in the real world. And in college, I learned that science could even be used to help people treat diseases and keep them healthy. And that has inspired me to do the work that I'm doing at Dartmouth today. At Dartmouth, I study breast cancer. You've probably heard of cancer before, but what is it and how does it happen? Cancer is made up of a person's own cells. A healthy person is made up of trillions of cells, and they work together to help your body function every day. Cells in your lungs help you breathe, cells in your brain help you think, and cells in your muscles help you to move. This teamwork is essential to make sure a person's body does everything it needs to, but sometimes cells can be rewired and stop working together, and this is called cancer. Cells can be rewired in many ways, including genetic mutations, exposure to smoking, or just old age. And one of the things that cancer cells do is grow uncontrollably, and in order for this to happen, they have to communicate with the healthy cells around them to tell them to move out of the way and make space for the cancer cells to grow. And my job is to study how the cancer cells and healthy cells communicate with each other to make those decisions. One way they talk to each other is through the proteins on the surface of their cells. Because we can't see cells with our own eyes, we have to come up with creative ways to see what those proteins are doing. And I'm going to take you to the lab and show you one of the ways that we look at these proteins, called a Western blot. So now that I'm in the lab, I'm doing the first step of a Western blot. After harvesting the protein from the cells, I'm loading the samples into a gel that has buckets that holds each of the samples. Once all the samples are loaded, we run an electrical current through the gel that causes the proteins to move down through the gel and be separated by size. As you can see, the dye moves down, and eventually it will completely disappear at the bottom of the gel. Once we finish running the gel, we can develop the membrane and actually see bands that show us the level of protein expression in the cells. It is with methods like these that my lab mates and I, shown here, get to use science to work together every day to better understand how cancer cells work and to strive to always improve treatment options.